I'll start again. So the presentation is about novel Spiro hybrids of two indolinone and thiazolidinone scaffolds as cell cycle checkpoints, pathway regulators with potential anti-cancer activity. Unfortunately, as we all know, that cancer is still a constant battle globally with a lot of development in cures and preventive therapies. And that globally, over the past several decades, cancer continued to be the second prime cause of death. According to the International Agency for Research on Cancer in 2021, around 18 million people were infected, 9.6 million people among them had died due to life-threatening cancer. The morbidity and mortality of cancer is expected to continue to grow for a long period, and cancer burden will rise to 30.2 million new cases and 16.3 million deaths by 2040. The high prevalence and mortality make a search for effective prevention or treatment for cancer really is an urgent medical concern. In recent years, cell cycle and checkpoints pathways regulations are offering new therapeutic approaches against cancer. As the control of eukaryotic cell growth and division occur at 3 critical points. These critical points are shown from the figure below, indicating the cell cycle. They occur at the G1, late G1 phase, G2M phase, metaphase, anaphase transition. These critical steps are also known as checkpoints, where at these checkpoints, there has to be balance between the growing promoting or the growth promoting pathways and the intrinsic suppressor pathways for the healthy cell division. Otherwise, cancer would occur. So, regulation to growth and reproduction involves many regulatory proteins, such as cyclin proteins, cyclin-dependent kinases, oncogenes, tumor suppressor genes in interphase, and mitotic checkpoints proteins. These allow the cell cycle to proceed or inhibit its precision. Our design of checkpoint regulators is based on hybridization of different pharmacophore from various bioactive substances into a single molecule. This hybridization strategy is really a potential weapon to prevent drug resistance and provides more leads with complementary activities and multiple pharmacological targets. The first bioactive pharmacophore is the 2-indolinone pharmacophore, which is clinically approved as anti-cancer drugs such as sunitinib, sorafenib, simaxinib, orantinib, ninantinib, and hesperidine. They all share the oxyindole moiety, and they are FDA-approved drugs targeting the cell cycle and checkpoint pathways. The second bioactive pharmacophore is the spiroindolinone. The key structure characters of this compound is the spiro ring fused with the indole moiety at the carbon-3 atom with different heterocyclic compounds. They act as active MDM2P53 protein-protein inhibitors. As also the thiazolidinone moiety really is a potential anti-cancer agent. So in view of the above facts, the current work describes the synthesis of novel series of thiazolidinone derivatives based on a hybridization strategy of the two bioactive moieties. Synthesis of the compounds started by reaction of the substituted isotin with benzyl bromide in anhydrous potassium carbonate, stirring at room temperature for 12 hours, leading to the formation of the N-substituted isotin. Then, formation of Mannich phases was done by reacting with different anilines in glacial acetic acid and refluxing for 6 hours. Then, afterwards, a mixture of equimolar amounts of the mannose bases and thioglycolic acid was irradiated in the microwave for 2 to 5 minutes in a catalytic amount of zinc chloride, leads to the formation of the targeted hybrid compound. This is the structure of the final hybrid molecule that is designed to act as a checkpoint regulator, hybrid of two moieties, the Spiro Thiazolidinone moiety and the oxyindole moiety, where the X and R were chosen to assess to optimize as much as possible the lipophilic electronic and steric character contributions. Structures and purity of the target molecules were confirmed by spectral elemental methods of analysis and were consistent with the proposed ones. Then, the in vitro cytotoxicity of the targeted compounds was evaluated using MTC colorimetric assay for 3 cell lines: human liver cancer, breast cancer, human colon cancer, in addition to normal cells. The non-cancerous cells were used to evaluate safety selectivity of the tested compounds. Doxorubicin was used as the reference drug. The activity of the tested compounds are expressed in IC50 as the value of micromolar and are given in table 1. From the given table, we can see that the selectivity of the doxorubicin wasn't good enough. It was about 1-fold. However, the selectivity of compounds 3E and 7E was about from 5 to 8 folds, and the selectivity of the other compounds was about from 9 to 16 fold selective to cancerous cells. And from the given table, we can see that 5 compounds show significant cytotoxic activity against the cell lines, such that compound 7E, 9E, 10E showed significant cytotoxic activity against Hep G and breast and colon cancer cell lines. Compound 7E showed activity against the breast cancer cell line, and compound 9E was more significant activity against the colon cell line, and compound 10E was more significant for the liver cell line. Compound 3E and 12E were similar. They didn't show any significant activity against the breast cell line. Then the most active compounds were tested for the expression of 4 key genes for revealing the possible molecular mechanisms for their anti-proliferative activity. These genes were P53, CDK1, and caspase and topoisomerase 2 enzymes. Doxorubicin was also used as the reference drug. Doxorubicin elevated the expression of P53 and caspase 3 by 5 folds, and then significantly caused reduction in CDK1 and topoisomerase by 50 to 60%. All the tested compounds, as we can see here, elevated the expression of caspase 3 by 5 to 6 folds, which is more than caspase, which was more than the shown by that of doxorubicin. Here we can see that compound 3E was unique and was the only derivative that reduced the expression of topoisomerase 2 enzymes by more than 55%. Compound 12E only had activity on the elevation of the expression of caspase 3 enzyme. We can see that compound 9E and 10E had similar gene profile, where they increased the induction of expression of P53 and caspase 3 by 5 folds, and made more than 50% reduction in the expression of CDK1. Compound 7E had similar profile, with exception of that they had no effect on CDK1 expression. Interestingly, compound 3E and 12E were similar again and directly elevated the expression of caspase 3 by 6-fold without affecting the activity of expression of p53, escaping all the upstream apoptotic events. This approach is very promising hotspots where direct targeting of the caspase 3 could help in managing resistant tumors. Then, the effect of the active compound 3E and 7E on the concentration of the topoisomerase enzyme was estimated. Compound 3E produced a significant reduction in the concentration of topoisomerase 2, which was comparable to and consistent with the effect on gene expression. However, compound 7E didn't show any reduction in the concentration of topoisomerase 2, and this was expected from the gene expression results. For all the compounds, the 5 active compounds, the concentration of the cleaved caspase 3 was estimated and caused significant increase in the activity of the cleaved caspase 3 by varying degrees from 300 to 500% compared to the untreated cells, in agreement also with their effect on gene expression. Finally, a molecular modeling technique was used to investigate the most appropriate orientation and interaction of compound 10E in the active site of CDK1, where compound 10E showed was the most significant activity for inhibiting CDK1. And from the docking score results and examination of the 2D and 3D pose revealed that it's fitting well in the binding site and displayed favorable interaction with crucial amino acid residues at the binding site of CDK1. So, conclusion to our preliminary study or our work, it aimed first to develop new indoline thiazolidinone hybrids targeting cell cycle checkpoints as potential anti-cancer agents. The design was based on hybridization of different bioactive pharmacophores to single molecule to enhance efficiency through multi-targeting, selectivity, and safety. Then molecular docking studies afterwards revealed the importance of both 2-indolinone and thiazolidinone moieties for CDK1 inhibitory activity. The design of molecule also showed multiple molecular targets in cell cycle checkpoint pathways. And this is preliminary studies, and then further biological activities will be performed to explain deeply the mode of action inside the cancer cell line. Thank you for your attention.